So welcome and thanks for attending this presentation from the Indiana CTSI Access Technologies program. Today's presentation is going to feature postdoctoral researcher Nick Morato, who's going to tell us about the high-throughput mass spectrometry facility for chemical synthesis and biological analysis. It's now available for use at the Bindley Bioscience Center at Purdue. This system can be used by Purdue researchers, but also by external parties. This high-throughput screening platform was constructed by the Cooks Group at Purdue in cooperation with DARPA, but recently, with the help of NIH NCATS, the system has been morphed into a groundbreaking new platform that is revolutionizing the way that medchem research is carried out. Nick has been at the center of these amazing new developments and is going to tell us all about it today. So without further ado, Nicholas Morato.
Thank you very much, Chris. And thank you all for attending the seminar. So in general, I'll try to provide an overview of what type of research and services can be carried out at the facility, but we'll get there through perhaps the history of this technology and how does it work. But before I go there, so just perhaps as a reminder, the high-throughput DESI facility is administratively under the metabolic profiling facility, which is part of the Bindley Bioscience Center here at Purdue. And the Bindley Bioscience Center offers a lot of different services in the life sciences areas here. There are a couple highlighted ones. In particular, right now, the high-throughput DESI technology is covered within the metabolite profiling facility. And so maybe by now, most of you will be wondering, what is DESI and why are we talking about a high-throughput facility? So we can start there, kind of that part that is the technological core of the facility. So DESI stands for desorption, electrospray ionization, mass spectrometry. Desorption electrospray ionization is an ionization method, a way to generate ions so that then you can perform mass spectrometry analysis on them. And that method was developed in Professor Graham Cook's lab about 20 years ago. So to start thinking about how to do high throughput experimentation using this methodology, we first need to overview a bit of how this method works. So DESI is pretty much a micro splash experiment. If you imagine the tooth flossers that you use, the water flossers that you use to clean your teeth, it's akin of that, but in a very microscopic scale. So you use a charged solvent spray to impact on a surface, and your samples are deposited on this surface. Then those droplets would generate, the first droplets to arrive will generate a thin film. In that thin film, a micro extraction event will occur, and in that way, you desorb the analytes from that surface, and the next incoming droplets through a stochastic momentum transfer events will eventually lead to the surface splash droplets that contain the analytes desorbed from the surface. And eventually those droplets will lead to traditional ionization mechanisms, dry ions that you will be able to analyze with mass spectrometry. And so DESI brings you a lot of advantages inherently through the mechanism that it has. So for instance, you are using a spray, and again, go back to the water flosser. You can choose which part of your mouth you're cleaning. So you have a spatial control over the sampling event, meaning that you can aim this spray wherever you want to analyze whatever area you want. You also have an online microextraction event that is happening, meaning that you can deal with very complex samples, as those samples are always deposited outside of your instrument, outside of your fluidic system. As you can see, it's a completely contactless method. You have clean solvent going through the capillary, and just the droplets with the desorbed analytes go through the instrument. So your complex, perhaps nasty sample, it's all deposited outside, and it's never going to be clogging or carryover happening through your system. So you can deal with a lot of complexity just because of how the methodology works. And that in particular is what had opened a lot of different applications for DESI technology. Perhaps the most well-known is related to molecular imaging. Again, as you have a spatial control over the sampling event, you can aim this spray and raster across, let's say, a tissue section, line by line, acquiring molecular information from each one of the pixels that you raster. And in that way, you can generate ion images from, for instance, in this case, a tissue section of a swine fetus. And so that experiment has been on for perhaps 2 decades now. And that's part of the technology offering that is within the facility services. But it's perhaps a bit more interesting to translate that imaging experiment, which is inherently slow as you have to raster one line by line and acquire information from each one of the pixels, to a faster experiment. So if you apply the same logic of analyzing a tissue section, but now you want to apply it to many, many samples very, very quickly, the only thing that you need to consider is just to start abstracting what a tissue is. So a tissue, in essence, is just a mixture of compounds with some sort of special distribution. But then there's nothing stopping you from creating your own arrays of mixtures of compounds with whatever special distribution you're interested. And if you do so, you can translate directly the logic of an imaging experiment to a high throughput experiment, in which if you can deposit arrays of samples like these little spots on a surface, then you could image that surface and get the molecular information from each one of those spots very rapidly in a very efficient manner. And that was the initial approach that we took, as Chris mentioned, when this system, this high throughput DESI platform, was built throughout the DARPA Make It. The idea was to deposit an array of samples, be able to pretty much image that array, and from the ion images that you get, be able to infer if a reaction was happening or not. Are you producing the product that you're expecting or not? And this worked pretty well. As you see here, a couple of actual examples from deposited reactions, and you can identify which ones of them work from the whole array or not. But it's a bit inefficient. Why? Because if you use the imaging logic over an array of samples like the one that you see over here, you will see a couple of drawbacks. So you are limited by how big is the area that you screen. If you put less or more spots on the same area, the analysis time is not going to change. Also, you're passing through the same sample multiple times by rastering these lines, and so you're acquiring repeated information. So what we did was try to evolve that same imaging logic to a more high throughput full-on experiment in which you have random access to any particular spot within the plate, because what you're doing is jumping from sample to sample, spending half of a second or so on each one of the sample and then jumping to the next one. Something as what you see in this video with a sort of random array of dye spots. And of course, that increases the speed, gets rid of all those drawbacks, and gives you random access to any spot, so you could perform a secondary confirmatory analysis on any one of the spots as you wish. This is a great idea. In practice, it's a bit more difficult to achieve. So to achieve this, we had to implement some sort of calibration methodology, because you need to know where those spots are so that you can actually jump from one to the other very quickly. And we achieved that calibration with a 3-die mark setup that we put near the corners of the plates that we use. And I'll go through the plates in a minute. And that allows us to identify where all the spots are and how to jump on top of all of them. But following that idea, we pretty much put together a bunch of custom and commercial software, robotics, and analytical instrumentation that gives you the high precision that you need to be able to perform this experiment and achieve the throughputs that we wanted. So here you see a diagram that tried to summarize a bit of the instrumentation of the processes of this platform. And this platform is pretty much the core of the high-throughput DESI facility within the Bentley Bioscience Center. We have fluid handling robotics, and with that you can manipulate or prepare any of your samples. Note that we don't use any sample purification or separation steps. This is direct analysis of the complex mixtures, precisely because of the capabilities that DESI brings to them. And those samples can be pretty much anything you can put on a well plate. So initially through the DARPA project, we only explore organic reaction mixtures. And later on, we explore more towards the biological space, so we can do all sorts of biological assays, and you'll see examples of that. But also other sorts of biosamples. So you can think of bacteria, you can think of tissue. Anything really that you can put on a well plate, we can eventually use it for. But you have the fluid handling robotics to do that sample handling. You have a robotic arm that takes plates from the fluid handler and puts them on the DESI stage of several mass spectrometers that we have coupled to the system now. And you, of course, have an overarching software control system that allows you to integrate these parts and also to automate the whole workflow. What you do with the samples is have them in standard 384 well plates. And then we use a pin tool to perform a very small transfer. So we transfer 50 nanoliters of each sample, few nanograms or sub-nanograms amounts of material from well plates to what we call a DESI plate. And this DESI plate is nothing more than a glass slide that we coat with a light layer of adhesive, and then we stick on top a PTFE membrane. And so we spot the samples onto that PTFE. We can spot 16 different well plates onto a single one of these DESI plates. So in the same area, the same dimensions of a well plate, you can actually put the density of 16 of this. You do the math, that's about 6,144 spots that you can put on a single one of these plates. So it's very high density arrays. These spots are about 800 microns in diameter. And so once you spot that array, and of course, for that, you need very high precision robotics, you perform the spot-to-spot analysis that you saw in the previous slide to jump from sample to sample and acquire information from each one of these. We can go up to spending about a third of a second on each one of the samples before moving to the next one. But overall, we try to use the platform at a throughput of about a sample per second. So you can do the 6,000 samples in a little over an hour. So all the data is automatically collected and also automatically processed. And you can, for instance, take advantage of interactive heat maps to see what type of reaction mixtures give you, for instance, particular products, or you can use all that information to perform quantitative analysis, which is a bit more interesting for the biological classes. I'll give you now a quick demo of the system through this video. So now we're looking at the deck of the fluid handling robot. We're having a plate of samples already ready. Now we're using the pin tool to transfer 50 nanoliters of each one of those samples to a Teflon coated glass slide to this DESI plate. So you can see in this case, we just do one transfer, but we can do replicate transfers from the same well plate or transfer up to 16 of those well plates to one of these ones. You don't have to use all the positions if you want to. Once the plate is ready, it's put in the back, sent to a shuttle. The shuttle sends it to the back of the robot where there is another robotic arm that takes the slide and places it on the DESI stage ready for analysis. Now you're seeing the screen of the computer connected to the mass spectrometer. This is the software that allows us to control the whole system. Before the analysis of any plate, the only step that we need to do is a manual calibration, which involves aligning the DESI spray with those 3 dye marks near the corners of the plate. And that's to calibrate the positions of all the other spots in the array. This particular demo is using 4 different 384 well plates. And so it's 1536 samples. You can define whatever settings for the mass spectrometry experiment you want to do. And here you're seeing live data acquisition from the platform. This is from a biological assay directly from the complex mixture, salt, non-volatile buffers, everything in, and you can monitor the substrate and the product of the reaction with very high quantitative performance. And here you see live how the stage moves. I hope that you can see that the stage is kind of jumping from sample to sample, and you're spending less of a second on each sample before moving to the next one to complete the analysis. And that's how the whole system operates. But now let's talk about what we've done with it. So we've tried to summarize all the capabilities of the system in these 2 classes. So one class is related to synthesis, one class is related to biological analysis. So for the synthesis part, a lot of the synthetic capabilities of the system rely also on another inherent characteristic of DESI as an ionization method. So as I mentioned earlier, DESI generates these secondary microdroplets that contain the analytes that were deposited on your substance. So if those analytes happen to be reagents of a particular reaction, let's say A and B, as those secondary droplets that contain A and B fly, what will happen is that an accelerated reaction will occur and you will end up with the product C of the reaction. And that product C is what you're seeing in the readout of the system and in your mass spectrum that you get at the end. And so you can very rapidly monitor reaction even without the need for a pre-incubation. Taking advantage not only of the throughput of the platform that you achieve because of the technology and the speed of the stage and so on, but also of this chemical phenomenon that allows you to screen reactions without the need for any pre-incubation. And that reaction acceleration phenomenon has to do with the interface of the droplets, which are very particular spaces where high reactive species can be generated. There are low pHs, high electric fields, and a combination of all these factors, partial solvation of the reagents lead to the acceleration of the reactions that we're pretty much leveraging completely to use the system. So how have we used it? Well, you can use those accelerated reactions and this very fast platform to directly screen your complex reaction mixtures to, for instance, identify what are the most optimal conditions for a particular transformation. And here are two examples. One is the synthesis of lomustine, an anti-cancer agent, in which, for instance, we optimize this particular nitrization step. And after screening it very rapidly with the DESI system, in an hour, you do 6,000 samples, identifying what is the best condition from 6,000 potential conditions that can include changing the solvent, the catalyst, the ligands, the stoichiometry, the order of addition of the reagents, everything you can think of. You can translate those direct reactions, those reaction conditions directly to a scale-up approaches, for instance, a flow reactor. And what you would see is that the optimal conditions found through the DESI platform, leveraging this reaction acceleration phenomenon, can be directly translated to scale-up approaches without any problem, giving you higher efficiencies and higher yields. Here's another example for a cross-coupling reaction synthesizing this drug candidate that is actually developed by the Syntim lab here at Purdue. Now you can also use the system and these accelerated droplet reactions to perform late-stage functionalization. So let's say that you already have a compound that has some biological activity and you want to do some SAR, you want to modify it selectively in one particular location to try to address how, for instance, candidates will behave with your biologics. So you can also do that with this droplet reactions and this high-throughput system. Here is just a couple of examples of late-stage functionalization of opioids. There is a new opioid receptor agonist, PCM21, which is experimental, and a very well-known new opioid receptor antagonist, naloxone. So those two are important in the generation of non-addictive but still analgesic opioids, and also opioid overdose treatments. So here are just two particular reactions. We perform many reactions with the systems, an alkylation reaction, a suffix-like reaction. And here you see typical results that you will see with the platform. So for instance, for the alkylation of PCM21, here you see the substrate of the reaction and the product of the reaction. And this is just by mixing all the reagents, spotting them on the plate, and analyze them right away. So you'll see a significant amount of product being formed in the millisecond time that these droplets fly. For the suffix reaction, the main product is the intermediate suffix, the suffix intermediate, but then you also see a little bit of the amine exchange at the end. What's also important with the system is that, as I mentioned earlier, you have random access to any spot. So for instance, if you see this result on any particular spot, you want to make sure that you're actually making what you say you're making, you can go back to that spot and perform a tandem mass spectrometry experiment. So you can select any of the products, break it apart, see the fragments, and see if those fragments agree with the structure of your proposed functionalized reagent. PCM21 is a very interesting case because depending of where you're functionalizing the drugs, you could clearly see trends in the tandem mass spectrum of the functionalized products. So this is the spectrum of just the substrate PCM21 without any modification. And if you start modifying one side, the phenyl side of the molecule, or the other, the thiophene side of the molecule, you will either preserve the product ion, so where these peaks are on the x-axis, the M over C axis, or you'll preserve the relationship between the peaks and the precursor molecule. That's called the neutral loss. Depending what you preserve, the neutral losses or the product ions, you can definitely assign that transformation to either the phenyl side or the thiophene side of the molecule. So you don't only get a reaction screening based on the conditions of the transformation, you can not only assess that if the transformation is successful or not, but you also get online structural elucidation using this platform with a very high throughput and without the need to deposit any more material. This is acquired coming back to spots that have been already screened to assess if the reaction works or not, and now just getting a structural analysis from those same nanograms of material deposit. Now, the third application is related to try to even leverage further those droplet accelerated reactions. Because for the previous two, what you're doing is sending those droplets to the mass spectrometer, getting a readout, and that's getting you information to know if a reaction is working and how well is it working, are you making the product that you're expecting, so on and so forth. But could you use those droplets for something else? Well, yes, you could collect those nanogram amounts of material if you can intercept those droplets somehow, and then use them, for instance, for biological testing. And so What we're showing here in this video is a very proof of concept type of example in which we're just putting a filter paper in front of the instrument, and we're using it to collect those droplets that contain the products of those accelerated reactions. Of course, it's a slightly slower experiment. We raster the spots back and forth to collect as much material as possible. But then if you analyze that strip of paper in which you deposited some sample, you will see that as you go through the area that has a spot, a lot of the product of the reaction shows up. So you're effectively collecting the product of these reactions in this strip. Of course, to take this from a proof of concept to an actual device, that has been a lot of work over the past couple of years, but we now have a prototype that pretty much does an array-to-array transfer using a combination of linear and rotary motions. So you have access to different positions on a line of the array and also to multiple lines of that array. And in that way, you can collect the material of, let's say, a reagent plate, obtain a product plate that then you can use for something else. And that something else is biological analysis. So that takes us to the other side of this box, to all the things that we can do kind of the biological route. So what can you do? Well, first, you can use this platform to perform completely label-free biological analysis. Why do we care about doing label-free biological analysis? Because most of the biological system, if you know color, no fluorescence, they have not inherent chemiluminescence. So usually if you want to use a plate reader, which of course is going to be very fast, it's going to do a parallel detection and so on and so forth, you're going to have to modify the native system that you're studying. So you're going to have to change the substrate, use a non-native substrate that will eventually give you a color product or a fluorescent product, couple a secondary reaction that can give you a detectable output, use radio-labeled compounds, and that of course increases expenses and the safety concerns around your assay. And so there are works around, but perhaps the most challenging part of using label assays is the interferences that you're introducing through your system to the one that you want to study by having to change it just to be able to get a detectable output. Now, relying on a mass spectrometry-based method, and in particular, in this case, SEMS, you don't have those constraints. Mass spectrometry is inherently label-free. It's a very sensitive, very high chemical specificity technique. And so if you combine that with the advantages of DESI, which is dealing with these very complex samples directly, you can actually analyze biological assays very, very quickly without any decrease in the throughput and without the need for any sample preparation or any modification to your native system of stuff. So we've done these assays with non-volatile buffers, so for instance, phosphate buffer, with a huge amount of detergent. So you think about tritone, a bunch of salt, etc., etc. Everything that the enzymes or any biological target needs to be happy and to work well, we've dealt with it directly without having to clean the samples, without having to separate or to purify in any way. And that gives you a lot of advantages. So just to show you some examples, this particular case, acetylcholinesterase enzyme hydrolyzes acetylcholine into choline. This reaction is carried out in phosphate buffer saline, a very high concentration of non-volatile salts, that perhaps with other mass spectrometry approaches, what you will do is just clog completely your system, be able not to spray anything, then you'll have to clean that for God knows how long to be able to get rid of the salt buildup. But we have no problem of analyzing thousands and thousands of these samples without even having to clean the instrument. And the most surprising part of it all is how quantitative information, how high quality quantitative information you can get from that sub-second measurement from this complex sample. So if you quench multiple aliquots of this reaction, and the quenching is just to fix exactly the time of the reaction, you will get results like this, in which you see a decrease in the substrate of the reaction and increasing the product of the reaction as the aliquots go by. And if you calibrate the ratios between substrate and product, you can get calibration curves. You can convert that to absolute concentrations. And with that, you can do any sorts of analysis that you want. So you can do kinetics on these enzymes. You can do Michaelis-Menten curves. For one of these curves, you will take thousands of samples, and we run all those thousands of samples of replicates to obtain very high precision data, as you see here. You can screen inhibitors for the enzymes and then characterize any inhibitors that you want, for instance, in vitro here through those response curves, or you can identify inhibition constants and inhibition mechanisms to more complicated and more sample consuming experiments. I think for this particular plot, we did about 4,000 or 5,000 samples, which sounds like a lot, but with the speed of the system, that is really perhaps under an hour of analysis. So it really brings a lot of advantages. Now, for acetylcholinesterase in particular, one of the projects that we're working on in collaboration with the Lawrence Livermore National is to try to address the problem of adduction of the enzyme, which is the way that chemical warfare agents act in our bodies, and try to reactivate the enzyme upon addition of some sort of reactivate. So we've shown that we can monitor that covalent inhibition. We can show that we can also monitor the reactivation process of the enzyme. And furthermore, that we can screen really a whole set of compounds for reactivators of this particular enzyme. Here's where I want to point out one of the advantages, or highlight one of the advantages of going completely label-free with this technology, and is that the standard way that you will use to study this particular enzyme is this thing that is called the elements assay. So to use this elements assay, which is a method, very old method that was developed to quantify titles, you have to have some titles to be able to use a method to quantify titles. And the way to obtain that title is to change the native substrate of the enzyme, which is acetylcholine, for acetylthiocholine, so that then you can obtain thiocholine, so that then you can add a reagent that can eventually lead to a colored species. And all this work is so that you can detect some sort of output from this reactor. We don't have to do any of this. We take acetylcholine and we monitor how much choline is made and how much acetylcholine disappears, and that's about it. And the problem is not only the work involved on doing this and how many more reagents and solutions, but also the interferences that it brings, in particular for the screening of reactivators of this. Reactivators tend to hydrolyze very efficiently acetylthiocholine. And so you will start getting a bunch of thiocholine, and you will start getting a lot of yellow color in your solution without reactivating your enzyme, just because the candidates that you're screening are breaking apart that non-native substrate that you're being forced to use to get a colored output. Now, if we use the native system of a study and we use the high-throughput DESI platform to screen this reaction, we get no hydrolysis because you're using the native system of the enzyme. And so you can screen oxygen at very high concentrations over very long periods of time. And then in this way, you can try to assess and discover new reactivators that can address, for instance, the resurgence of new chemical warfare agents. And that is not really the only enzyme that we work on. Over the past couple of years, we've really extended this methodology to 10 or 20 different enzyme classes. Here you just see a couple of them, just to give you a couple of examples of what we've done with them. So for instance, if you're interested in the antibiotic, antibacterial world, we've explored beta-lactamases. We can monitor the hydrolysis of the beta-lactams over time. We purify enzymes directly from microorganisms. In this particular case, we're actually leveraging the amount of salt that you have available in solution, and we're detecting some of these analytes as sodium adducts with very high sensitivities. A very similar approach we follow to study phospholipidases. This is in collaboration with Professor Andrew Mesecorp in projects related with Alzheimer's. We're detecting the formation of this lipid as a very high intensity sodium adduct with very, very high sensitivities, and then we can monitor what the enzyme does over time, get kinetics, affinities, etc. We can also do, for instance, substrate scope type of studies. And you can imagine this in one application might be direct evolution of particular enzymes. So we can do substrate scope studies. In this case, for a methyl transferase, we can assess if you're making the product starting from different substrates. And one case that we like a lot, and that I come back at the end to kind of show you a very big research project example, is this case about sulfur transferases, in which we're quantifying directly the amount of esterate sulfate that is being formed by the action of the enzyme. Here you see an example of a calibration curve. I've mentioned before that we get very high quality quantitative performance, so I need to show you some proof of that. This is a calibration curve of thousands of samples run over many days, perhaps many months. And you can see the high performance that we get. The inset shows a low concentration range, and we get limits of detection in the order of 60-ish nanomolar for this particular analyte. If you do the math, considering we're depositing 50 nanoliters of solution, that's low picogram going down to femtogram levels of analyte actually deposited on the surface. And using this type of quantitative performance, we can again study kinetics of the system. One of the advantages that you have, again, for going completely label-free is that if you change the substrate of the reaction, you don't have to start from scratch developing a new method that will eventually give you a plate reader output. You can just change the substrate and keep detecting whatever the product of the reaction is. So here are two examples, pregnenolone and cholesterol Michaelis-Menten plots. You can see very high quality data, and we didn't have to do anything. We started with cholesterol, we move on to pregnenolone, then we move on to other esterate substrates for this particular enzyme without to have to change anything to the method that we were using. So that's another advantage of the label-free high-throughput technology. Now, in some cases, especially when you use an internal standard, you get an extra advantage, that is that you really don't need a calibration curve. If you're not interested in, for instance, actual rates or actual concentrations, we can get affinity parameters directly without a calibration, which is an advantage in cases where it's very difficult to obtain the product of a particular reaction. But the value, the affinity values match exactly what you will get if you calibrate it. Another advantage of using mass spectrometry for the study of these biochemical systems is that sometimes you get more information than what you were asking for. So for instance, if you take this inhibitor of the enzyme and you do the biological assays, yes, you can get how strong it is, you can get an IC50, you could get a KI, etc., etc. But even through this simple dose response experiment, you're going to get more information than what you were asking for, which is just how strong is this as an inhibitor, because you're collecting all the spectral information. And so you kind of start seeing things that you might not have expected. For instance, this other peak, as you go down here in the spectra, you're increasing the amount of inhibitors. So these spectra are just corresponding to different points in this concentration, in this dose response curve. And you will see that the product of the reaction, which is cholesterol sulfate, here has 465 decreases. It makes sense. You're inhibiting the enzyme. This 395 peak is the internal standard. So that's what we're using for quantitation, not changing, all good. But you start seeing this other peak at 467 coming up and coming up and coming back and coming up. And you might wonder, what is that? Well, it's the sulfonated version of this inhibitor. So with a single experiment, you don't only get quantitative values of how strong is this inhibitor as an inhibitor, you're also directly getting the mechanism of inhibition. There's a competitive inhibitor that is getting sulfonated by DESI, and all that without really having to do anything else. So that's another advantage of using. But enzymes is not the only thing that we've done. If you're not interested in enzymes, sorry that I bored you for the past 10 minutes. But for instance, you might be interested in receptors. So if you think about receptors, we have to measure binding affinities, for instance. And we've developed, in particular, competitive binding type of assays, in which we've incubated membrane-bound receptors. We're very interested in opioid receptors through collaboration with NCATS and other groups at Purdue, at the University of Buffalo, and MIT. So we've developed these competitive assays in which we incubate membrane-bound opioid receptors with an in vivo ligand, leucine encephalin, and whatever drug you're trying to assess. And what we do is, after an incubation period and get to reach an equilibrium, we measure the amount of leucine encephalin or the competitive ligand that you want to use that is free in the solution. So all this happens automatically. We let the reactions incubate and we just have a single filtering step using the fluid handler. And then we measure how much of that competitive ligand is free in the solution. If you have a lot of that competitive ligand free, that means that your drug is very strong because it displays a lot of it. If you have very few amount of that competitive ligand free, then your drug is weak because it didn't displace much of the other ligand. So we can with very high sensitivity, quantify that competitor using an internal standard. Here we're using a methylated version as the internal standard. But what you will get is that data matches pretty well the trends that you will expect for binding affinities for these compounds. So we've done this with multiple opioid receptors, mu and delta, different drugs. So here you guys see aspirin, you almost displace no leucine encephalin because aspirin is not a good opioid. But if you start using fentanyl, you'll see a lot of displacement. Fentanyl is a very strong opioid, and so on and so forth. We've combined the system with other sort of digital virtual resources, part of this binding assay label-free methodology. Won this NCATS Aspire Reduction to Practice Challenge. It shows the advantage of using high-throughput label-free mass spectrometry to perform these biological assays for this particular system, at least. If you're not interested in enzymes or receptors, maybe you might be interested in analyzing many tissue samples very quickly. And that can be done for many reasons. One is, for instance, biomarker discovery or validation of biomarkers with large cohorts of samples. Imagine like a tissue bank. And so the use of high-throughput platform, of course, inherently not portable. The system is quite big, as you saw in the video, but you can use it to support applications in other areas. For instance, intrasurgical applications, where you put a mass spectrometer inside the operating room. Well, somehow you need to know what are you looking for with that mass spectrometer in the operating room. What biomarker are you looking for? What type of classification models you use? And if you want to build robust classification models or to identify robust biomarkers, you might have to screen many samples. And you can achieve that with this platform very quickly. So for instance, we can take tissue biopsies from directly the surgical room or from tissue banks, put them on a wall plate, and without doing anything, so just frozen biopsies, transfer nanogram amounts of material to one of the DESI plates, screen it with the same speed that we do everything else, and get molecular information from it. So we can, for instance, monitor particular biomarkers, in this case, 2-hydroxyglutaric acid, and we get 100% accuracy identifying the mutation of the isocitrate dehydrogenase enzyme in brain cancer biopsies. You can also use it for discovering new biomarkers. So you can go through a completely untargeted analysis and try to assess what type of lipids or metabolites, and here I want to highlight that we focus mostly with the DESI technology on the small molecule side of things, or mostly lipids, metabolites, and small peptides. You can assess which one of these small molecules are indicative of having more tumor cell percentage than the others. You can correlate that with pathological readouts, or et cetera, et cetera. You can combine the idea of using enzymes and tissue. So for instance, you can monitor how drugs behaves directly on tissue, or you can monitor what happens on different tissues if you spike a substrate. You can start following full metabolic pathways and start seeing how a substrate degrades, intermediate products appear, some other branch pathways start taking place in different organs, and so on for this type of metabolic applications. We've also demonstrated you can use bacterial samples. So either you can grow bacteria in liquid media or well plates, and we can use that directly. You can also grow it in agar plates. We can pick colonies from those agar plates, transfer it to well plates, do the exact same analysis. You can classify a species using this small molecule profile with very high performance. Here you see a couple of examples of different sets of bacteria. You can even go down to see what it's separating them from everything else. In case of Pseudomonas aeruginosa, a lot of quorum sensing metabolites that we're getting. We've also done it with yeast species in this particular case. Here you see a summary of identifying 30 different species of microorganisms. It's just a confusion matrix with different machine learning models. You get pretty much very high accuracy, close to 100% accuracy on the classification of these species. Now, we don't have to pick the colonies either. So if you take a picture of your agar plate, we can use some image processing to identify colonies. Then we can use travel salesman type of heuristics to identify the short path route to each one of the colonies, and then just jump from colony to colony, getting molecular information from each one of them. And for instance, we could tell you which one is the best producer for something. And then you can pick that colony and continue, for instance, a directed evolution process. And we can couple this analysis. This is just an example with bacteria, but in general, with other more new mass spectrometry scan approaches. So this is a new scan that we've developed in the lab, which is called two-dimensional tandem mass spectrometry, in which you acquire all the information from all the product ions coming from all the precursor ions in your sample in just a 1-second scan. So if you imagine here, you're combining throughput over throughput over throughput. You have a system that allows you to automatically handle samples, screen them very rapidly without any sample preparation at about a second per sample. And in that second, you're getting all the product ion information from all the precursors in your sample. So the amount of data and the density of the data that you get just exponentially multiplies. So those are just a recount summary of multiple different applications that we have with the system, both in the accelerated synthesis side and the bioanalysis side. But if you start thinking a bit on kind of drug discovery, you can combine all these applications. So if you have a way to screen reactions, find which reactions work better, then you can use the system to generate nanogram amounts of material, and then you can test the biological activities of those materials. You pretty much consolidated this drug discovery, and in particular, the early drug discoveries. Because if you think of drug discovery as a very long and complicated process, but if you just focus on the early drug discovery part, you will see that it relies very heavily already on high throughput experimentation. You need to find a way to screen biological samples, such as tissues, so that you can identify biomarkers and then potential biological targets. And we can do this with the platform, as I just show you in a minute. You need to be able to rapidly synthesize thousands of compounds so that you can test them. I show you that we can do that synthetic part with the platform. And you have to be able to test the biological activities of those compounds very rapidly again. And I'll just show you many examples that we can do that with. So if you put all those activities together, you're really consolidating this early part of the drug discovery funnel in a single technology on this single system, which is the core of the facility. And so maybe it sounds a bit too good to be true. So let me show you actually an example. And let's walk through this funnel in the last couple of minutes that I have. So this is a story that is started with the DESI technology. So we're using the exact same approach, exact same core technology in which the system and the facility relies on. But this started about 10 years ago. So it started with an imaging experiment. We have access to imaging experiments, though they're slightly slower, but we could do the exact same thing with the tissue microarray kind of high-throughput approach that I just showed. And what happened is that through these untargeted experiments, it was found that in prostate tumor sections, there was a huge accumulation of cholesterol sulfate that is pretty much almost absent in normal prostate tissue sections. And that led to a decade of research at Purdue and other places in which also using the DESI technology, you can do co-localization studies, identify that you don't only accumulate this metabolite, but you also overexpress the enzyme that produces it. And if you knock down that enzyme, you induce the death of prostate cancer cell lines, which indicates that this particular enzyme, the sulfotransferase 2B1B, is a new target for prostate cancer treatment. There's a potential new target for prostate cancer treatment, which is it's been quite understudied. So what do you do now? Well, you identify a target using this DESI technology. You can rely on the exact same technology now to characterize that target. You want to know how it works. You want to know the affinities, the kinetics of this particular enzyme. So you can do that with this particular system. Here you see examples, kinetic curve, different Michaelis-Menten plot with all the in vivo substrates of this enzyme, and assess the affinities, et cetera, really characterize this enzymatic system, avoiding having to develop whole new methods based on, for instance, a couple enzyme fluorescence reaction, directly just measuring the amount of esterase sulfate that is being formed by this enzyme. And in that way, avoiding a lot of interferences that comes from adding other enzymes that might get inhibited, and that you start having conflicting results when you're trying to find inhibitors for the enzyme that you're interested in. So once you've characterized the target, the next step in the drug discovery workflow would be to actually try to screen compounds. And here you can, of course, put all the organic chemistry that you want and generate new compounds and screen them. In this particular case, I'm just showing results of the screening of already libraries that are available here at Purdue. And so we can validate the high throughput screening methodology. We get very, very good Z factors or the 0.8 for this particular enzyme. Here you see results of all the validation experiments. You really don't have to do any math to be able to tell apart when something is inhibited and something is not. You see here in these boxes, but of course you can do the math to prove that you can do it very, very efficiently, very consistently over thousands of samples. And using this approach, you can screen thousands of different compounds against this particular enzyme. Here you see results of a 12,000-ish compound screening campaign. We've screened several thousand more compounds for this enzyme. This library didn't give us any hits, but the new library did. So we have a couple of hits for this enzyme, and we're very interested in those. But even if you've done, this is just a kind of a toy example to show what we can do next in the drug discovery workflow. Using galanterin, which is a known inhibitor of this enzyme, is not interesting because it's not a specific, but we can use it as a test sample. So if you find galanterin as an inhibitor used throughout your screening, then you can use the same platform to confirm that hit in vitro, which will be your next step. So you can do a dose response curve, make sure that it's inhibiting your enzyme, get IC50 values, if you want to go to mechanisms and KIs and so on. But then you can take it even further. And you can validate those hits not only on your purify enzyme system in your buffer and so on, but directly on process-cancelled cell cultures. So you can take process-cancelled cells, incubate them with the inhibitor, and directly from the cell get those response curves that validate your inhibitor directly on the biological process. That is really the power that we see on this platform, that it can consolidate all these activities of the early drug discovery workflow. Of course, you can work and you can have projects on either or these sites through the facility, but all these capabilities can be consolidated around the same technology. And based on that, and that's something that Chris highlighted, we've worked with NCATS recently to develop new technology that it's right now functional and available in the lab, based on pretty much this exact same platform, but a more consolidated new generation high throughput system that will be operated from NCATS in the future. That whole set of technologies and methods and experience is available to researchers, not only at Purdue, but all sorts of external researchers. We have collaboration with industry folks, with people in national labs, in other universities. We have right now through this facility about 20 active projects. We have several thousand hours of projects since this facility started about a year and a half ago. And we'll be very, very interested to see if we can help with any of the problems that you guys might have. And with that, I'll finish. And if you have any question, please feel free to unmute yourself or ask them in the chat box. And thank you very much for the opportunity to share this technology with you all.
Thanks, Nick. Great job. That's really amazing. Let's take some questions from folks online, if we could. Go ahead and type into the chat, or Jill, can they also just unmute and ask a question?
Yes, absolutely.
Okay. Let's start out while people are figuring out what they want to ask, but you've worked with a lot of companies. You can do so many things that it's a little bit overwhelming, right? It's high throughput analysis, it's synthesis, it's enzyme characterization and kinetic profiling. How do customers usually come to you? Do they start out with high throughput analysis, or it varies? Does it start with one thing and then grow into these combined uses that you described? What's sort of a typical pathway for a customer getting involved?
Absolutely. Yes. Usually we start with projects with perhaps a narrower scope. We tend to do first like a test in terms of, like, give you a case of biological assays, which is pretty much most of the projects that we work on at the moment. So it's usually people that have had trouble with any other kind of traditional methodology, interferences, developing a whole label method, and so on. We
label interference problems, things
label
like
interference problems, all these issues. So we get their systems, try to assess the quantitative performance that we get for those particular systems, and then we move on to characterization of the targets or whatever type of application they want to do. They usually start kind of narrow in that case of can we characterize this particular target, and then is that they evolve to a more large drug discovery kind of a screening campaign. So they usually start narrower, but in many cases they have evolved pretty big.
And so there's oftentimes a method development portion of the study, and then you go into your high throughput portion of the study. Is that correct?
Absolutely, yes. So initially we do the method development to kind of assess the quantitative performance. Once we have the performance that is satisfactory for whatever we're trying to screen, we move on to the larger characterization campaign or the larger screening campaign, and of course we do all the controls that we'll require. So we're doing a screening campaign. We will run all the inter-day controls, all the plate assays, so that we can assess C factors for that particular screening campaign, and so on.
And if people want to, if they have questions and are interested in, talking about running a study or sending samples or whatnot, do they contact you directly? Do they reach out to the Binley Bioscience area, or what's the right way to proceed with that?
I would say the fastest way, they can contact me directly for sure. But if you reach out to Binley, definitely they will eventually route you to us, so we can eventually sit down and talk about projects.
Is it okay if I put your email into the?
Yes, please do so.
Okay. All right. Online, do we have some questions for Nick?
Yes. Hi, Nick. Yeah, thanks
Hi.
so much for your presentation. Yeah, this is Li Fan from Chemical Genomics Core RUSM. So I do have two questions. My first question is, do you have a HPLC system connect to your DESI mass system? And my second question is, what about the cost for like screening one plate, like 384 plates? What about the cost? Thank you.
So we don't have any type of chromatography, any type of separation, sample purification, anything that goes before the DESI analysis. And that's part of the advantage of the technology, that you can deal with the complex samples without the need to have to pre-purify or separate or try to reduce the complexity in any way, shape, or form. So there is no LCMS involved. The samples are spotted on displays and directly analyzed using DESI. In terms of the cost, so there are different rates. I guess they're internal and external rates, and we can send you more information about that. I'm not super familiar exactly with the cost per plate, but the cost involves usage time of the mass spectrometer. We have different instruments coupled depending on the instrument that you want to use. If it is a low resolution, a high resolution, more sensitive instrument, I think the cost is a bit different, and just the plate. So the glass coated, the Teflon coated glass slide is pretty much the only consumable within system. So there is usage time, the plate, and if there is any like intense work required, then it's also the kind of work hours of the personnel of the facility. But in general, it's just usage of the instrument and then the plate. So if it is just one plate, it will be just that.
Okay, thanks. One more question. So is it possible to hook up a LC system to your DESI mass spec?
No, and there is no need. So the LCMS will only slow it down.
Yeah.
And you really don't need to do the separation beforehand, and that's really the whole point of the technology, and the way to achieve the throughput that we do is precisely not having that separation step. So yeah.
Yeah. Okay. Thank you.
Sure.
I think you make a good point about the, at a throughput of 1 sample per second, and it costs certain to get an hour's worth of instrument time, but you can do quite a few samples in that hour at 1 sample per second. So that's pretty amazing. Other questions for Nick?
I have a quick question. Hi, this is Cheetah Das from Purdue Chemistry. Can you hear me?
Yeah.
Okay, so this is amazing. It's really fantastic stuff. Thank you for presenting this. So we have, I might connect with you offline after this, at some point the next week. But I was really impressed with the sulfotransferase activity assay. So one of the enzymes in the NIH-funded project we have right now is they're basically nucleotide hydrolases. They break down either some form of ATP or GTP, and so but they don't tolerate any kind of modification on these nucleotides for like a classical plate reader kind of assay. So this would be great for us to come back, because when you break down the nucleotide, the enzyme catalyzes the cleavage, we can see clearly a different product, right? So your mass spec would be absolutely essential in quantifying and monitoring those products. So I can clearly see an example where we can't do really any labeled assay with that sort of nucleotides, and where this DESI would be some awesome. So yeah, and the other thing I have to add is I'm sort of thinking of working with you instead of working on a recharge. We pay for the plates and all that, but it'll be great for you to involve you some kind of a grant write proposal where we can develop these methods of looking at these enzymes, and they're sort of developing high throughput assay. So
Absolutely.
yeah, this is awesome. Thank you.
Yeah, be more than happy to talk with you whenever.
Okay. It sounds like a perfect fit.
Yeah.
Sounds like a perfect fit for the project. And it's not rare to find these instances where any attempt to put in labels or handles just gets everything screwed up, and you go down false roads and so on. So it's a real true advantage of the current technique. Other questions for Nick in the remaining time? I see we have Professor Cooks on. Graham, would you like to say a few words about this system? Or
I'll just make a comment in regard to costs. They're Midwestern costs, not California costs.
Yes, thank you, Graham.
Very
That's-
cheap. I mean,
That's very important. Yes.
We have a fire sale going on.
I think it is. I mean, the technique is so cool. We'd love to, do it with cost, of course, right? But
Yeah.
But I see sort of that the best way for me, at least, is to incorporate this in a proposal working with Nick and you, and
Sure.
to really, to develop this beyond what we can just do as a standard core facility kind of approach. Because
Right.
we can sort of,
as Nick said, there's a second system that's just been built, so we're actually not restricted in terms of instrumentation.
okay, that's great. Thank you.
Nick, I had a question. I think if somebody had their pet enzyme and they had their substrate in the product, probably not much time for you and your team to figure out, yes, we can turn that into a high throughput DESI assay to characterize that enzyme system, and then we can proceed with like enzyme inhibition studies, right? That turnaround for that is probably fairly fast. Or
Yeah, it is. I mean, we've had cases that they give us the material, and that afternoon we know if we're going to proceed.
So it can be a 1 day and up and running type of thing.
Yep.
So that's like huge advantage for medchem exploration. What about a company then has a library of compounds that is proprietary that they want to bring in for screening? Are you able to handle that and to keep those compounds sort of safe and secure? Is that a bridge that you've crossed here with the system yet? Or?
Yes, I think we have an agreement with one company through scientific exchange, and so we've definitely set up kind of those type of formal agreements through Bindley.
Right.
I perhaps not the best one answering how that whole process happened, but we've definitely done it before with a company.
All right, that's important to know. I'm going to turn it back to Jill to close, but thanks again, Nick.